more about turtles, more than we do every day in lab. So it's good for us. Yeah, so today we're going to take you through just a little bit of an update as to where we're at with turtle research. So that's going to be kind of the first 2/3 of the talk. It's just more of a review of what's been going on in the field recently. And then Paul is going to finish talking a little bit more about the specific techniques and research questions that we have as a lab at Hopkins. So yeah, my name's Will. I'm a 4th-year PhD student in the Beaver Lab in the Center for Functional Anatomy and Evolution, which is kind of a mouthful, but that's where we're from. So I'm a PhD student. Paul has been with us for a few years now. He finished his final year of your Hopkins undergrad with us, and then he completed his master's degree with us too, working on turtle research, and he's now sticking around for another year as a research assistant, both for our lab and for the pathology department. Okay, so just a brief outline of what we're going to talk about today. The first thing we're going to cover is the question, what are turtles? Now, this sounds like a kind of a basic question. I feel like as herpetology enthusiasts, we would all have an answer for that, but actually it's a question that has been controversial for many years in turtle research, and so we'll get a little bit into that and what I mean by that. We're then going to talk about what we've learned from the turtle fossil record in the past roughly 20 years, because a lot has changed. We found a lot of cool things out about ancient turtles based on some relatively recent fossil finds from the field of paleontology. And we're going to just briefly go over what we're doing as researchers using studying the developmental biology of turtles to try and get at some evolutionary questions. And Paul will cover most of that section. So this question seems kind of obvious, pitching this to a herpetology club, but actually it's kind of complicated. We all would say that turtles are reptiles, and that of course would be true. But actually, as evolutionary biologists for the past, well, it's kind of a debate that's been going on for almost 200 years, is exactly what kind of reptile that we know of did turtles evolve from? Of course, reptiles are extremely diverse. We have a turtle here, a crocodilian, lizard. Of course, birds are within reptiles. Birds are reptiles, snakes, and of course the tuatara. So they are diverse, both ecologically and behaviorally. And one of the most important questions that reptile research has been undergoing for the past couple hundred years actually is trying to figure out exactly where turtles fit within this broader radiation of reptiles. So we can go through a few of these hypotheses. This is a phylogenetic tree of amniotes. So this includes mammals represented by the mouse and reptiles on your right. And so one of the earlier hypotheses for where turtles fit within amniotes was actually outside of the clade or group that we consider and then we call reptiles. And so the idea was that turtles were descended from this extinct group of animals, and they were called pariasaurs, and they're part of this group that, if you read the literature, they usually come out of this group called parareptiles. And you can see they don't look much like turtles, but there are some similarities. They're kind of squat animals. That's a mount of the animal Scutosaurus from the American Museum of Natural History. They're kind of low set. Their limbs are pretty short. Their necks are pretty short, so kind of turtley. But really, what people were using to push forward this argument was that these animals had these big bony scutes in their back. And in these earlier times of turtle research, people believed that the shell was actually formed by, at least in its entirety, was formed by these bony scutes. So they were finding these animals in the fossil record, and they were saying, well, this kind of makes sense. They've got bones in their back, just like turtles do. Another hypothesis, and this is a little bit more modern than the prior one, and this is usually the outcome that we get when we do something called a morphological analysis, where we take physical characteristics of animals, basically score them in a giant matrix, and we get the computer to figure out which arrangement of relationships is the most realistic. And usually what we get when we run one of these analyses is a close relationship between turtles and lizards and snakes, or lepidosaurs, as we call them. Now, the most recent and the one that is kind of prevailing right now is based on genomic data. So prior to our ability to sequence and reconstruct the genetic code of animals, we were really relying on morphological data, and we still do to some degree. But now that we have a more objective means of assessing evolutionary relationships, most people accept now that turtles are actually more closely related to archosaurs, which is the clade that includes crocodilians and birds. But this does pose an interesting question and, well, interesting problem for us, and that is that our morphological data, which has been useful for all kinds of different other animal groups doesn't seem to be working as well for turtles. And so you can see that kind of being as a kind of an awkward problem for scientists, but actually it raises an interesting question. Why is it that we can't accurately reconstruct turtles from their anatomy? And the answer probably is that they might just be too strange for us to properly compare with other reptile groups. So we'll just go over a little bit about why turtles are so strange and so different. These are all reasons why, of course, we love turtles. Turtles, of course, have the shell. This is what they're most famous for sporting and presenting. They have what we call the carapace, which is the dorsal or the back of the shell, and the plastron, which is the front or like the belly plate. The carapace is formed by the vertebrae and ribs, which is something that the people who were supporting the pariasaur hypothesis didn't fully comprehend, and so it kind of was a red herring for them. So one thing that's extremely strange about turtles, if we have up here on your left, the kind of normal amniote configuration, it's the one that we have too, is our ribs are actually pretty delicate, narrow, there's space between them, and actually that space is filled by muscles called intercostal muscles. And then when we're exercising and when we're breathing heavily, those are the muscles that actually move our ribs to help our thorax ventilate air through our respiratory system. Turtles seem to have lost these muscles entirely, and the space between their ribs has basically been obliterated. The ribs expand and contact one another to form an immovable, akinetic basement to the shell. So that's one way that turtles are different to all other amniotes that we know of. I just talked about that. Okay. Yes, okay, so we'll do that again. That is also true. That's what I was talking about here. The first slide, what I'm talking about here is that turtles have a much fewer vertebrae than most other amniote lineages. When we look at ourselves, we have roughly 20 vertebrae. Turtles usually have only around 9. So they actually underwent a shortening of their thoracic vertebral count. We just talked about that. I just got them mixed up. Another thing that turtles do that almost no other amniotes do is that their ribs do something that we call axial arrest. And essentially what that means is that unlike in our anatomy, where our ribs leave the spine and wrap around, communicate with the sternum in the midline, turtle ribs just come straight out, which is super weird. And it leads to this condition where their shoulder bones, so if everyone feel for your scapula, which is this flat blade-like bone outside of your rib cage on your back, communicates with the arm. It's actually found within the rib cage of turtles. So this is a radical anatomical shift and super strange and hard to characterize as evolutionary biologists. Another thing that turtles do, this one's it's kind of a little bit more difficult to think about, but they do this thing called, they have flabellate ribs. I'm not exactly sure where that word comes from. We just use it. But essentially what it means is that when the ribs leave the spine, like they do in everything with the spine, they kind of spread out, kind of almost like a palm leaf. If you hold your hand up kind of like that, they spread out like that, as opposed to coming closer together and wrapping around thorax as they do in most other organisms. We call this the flabellate rib morphology. Turtles, of course, have the plastron, which is, again, very, very different from most other amniotes. Again, we have a sternum, that's this flat plate-like bone in the midline that our ribs communicate with. Turtle ribs, of course, don't do that, and they appear to lack a sternum entirely and have actually evolved this entirely new way of protecting their belly, which is the plastron. And this is derived from, at least we believe it is derived from, these bones that exist in many reptiles, which are called gastralia. These are sometimes called the belly ribs. Crocodilians have a lot of these. So turtles took those pre-existing belly ribs, expanded them, and formed a plastron out of them during evolution. The cranial anatomy of turtles is also extremely strange. Usually we characterize a lot of amniotes by, well, we can characterize many amniotes by the number of holes in their skull, or we call them fenestrae. And so mammals, including us, we have a single hole in our skull, and we call that the synapsid condition. Most reptiles, turtles excluded, frustratingly, have 2 of these fenestrae. We call that the diapsid, di for 2. It's the diapsid condition. Turtles don't have any holes in their head. The little one back there doesn't count, that's the ear. And then the one in the top left corner of the skull is just where the jaw closing muscles fit. So it actually doesn't count as a fenestrae. So turtles, it's also difficult to categorize them by the anatomy of their skull. So turtles are, of course, extremely diverse. There are more than 350 living species. Here's just a sample of how strange they can get. On the left is the mata turtle. I guess many people probably here will have heard of it, not from it. These guys are super strange. If you haven't watched videos of them feeding, they basically feed. If you've ever watched like a goldfish swim around a tank, it encounters food. They open their mouths very quickly, and basically the pressure differential between the water and their mouths sucks in the food. So mata do the exact same thing, and their skull is extremely weird. So if you're looking for something to do after this, I'd recommend Googling the mata skull. They're very odd. All the way on the left, your right, we have, of course, the alligator snapping turtle, large predatory turtle. If you guys came to Turtle Week this year, there was a big alligator snapper in a tub, which was cool. It's my first time ever seeing one. It was very, very cool to see. Yeah, right. Where's the snapper? Yeah, it's in the car, right? And then in the middle, of course, we have the famous Galapagos tortoise next to a horse, just to show how large these animals can get. And I actually didn't know this fact until I was building these slides recently, but Darwin himself, of course, the author of The Origin of Species, actually took one of these tortoises back with him from the Galapagos to England. He named it Harriet. She lived in England for several decades and then traveled with one of Darwin's friends to Australia. And she died relatively recently in 2006 in the Australian Zoo under the care of course, the Irwin family at 175 years old. So these animals are really incredible in their lifespan. And if anyone, I'm sure several people here have pet turtles, make sure you have a plan of something to do with them. Relatively, keeping your lifespan in mind, because these things can live a long time and you don't want to abandon them. Turtles, of course, are extremely tough, and that's down to an aspect of their biology known as physiology. It's the operation of their cells within the body. Turtles can actually slow their metabolism to the point where they only need to beat 1 to 2 times per minute. These are some just basic pond turtles that we could find in the northeastern United States. And if you can walk around enough in winter, you find a nice clear lake, you can see them sort of hanging out under there. And this is a state that we call brumation. So these are tough animals. They can adapt relatively quickly to pretty tough environmental conditions. And that is likely why they basically emerged almost unscathed from the KPG extinction. This is the extinction that wiped out 75% of all life on Earth. Turtles, as far as we can tell, only experienced about a 12% biodiversity loss over that extinction. So these animals pre-existed the dinosaurs, lived along with them as the dinosaurs evolved, and then outlasted them almost entirely, of course, aside from the avian dinosaurs. So it's kind of intuitive to think about why that extreme physiology might have helped them in such a destructive event that could shift climatic patterns and all kinds of things that many animals wouldn't be able to deal with. Turtles are also, of course, ecologically very diverse. They've adapted to live in all kinds of environments. There's a desert tortoise there. These things live in the deserts of Western North America, where temperatures can reach 140 Fahrenheit. They spend most of their time in burrows to avoid the heat, but they only need to drink a couple times a year. So again, that's that amazing physiology coming into effect. All the way to the relatively deep ocean or the open ocean. There's a leatherback turtle there. And then in the middle, we have the context that we're used to seeing turtles in northeastern North America, where they're just kind of hanging out by a pond, basking, looking for food and things like that. So it's a broad range of habitats that turtles can live in. So one of our jobs as evolutionary biologists is to find some way to characterize, well, to group and to organize all of this diversity. And nowadays we do so by their evolutionary relationships. And so we recognize 2 main groups of turtles nowadays. We have the cryptodiers, which are sometimes called the straight-necked turtles, and pleurodires, which are the side-necked turtles. And that description makes sense when you see these images. Pleurodires, they have this kind of serpentine S shape, and that's how they retract their neck. And cryptodiires, which retract it basically just in a straight line, just by their spine. And what's cool about this is actually this split relationship between pleurodires and cryptodiires is something that biologists actually figured out before genome sequencing. And once we double-checked with DNA sequencing, it created the same phylogenetic pattern. So we got it right without DNA, at least within turtles. And we call this a crown clade, some terminology that we use. And crown clades are defined by two extant lineages. So if Pleurodires went extinct, there would no longer be a testudine or turtle crown clade. And that's how we define crown clades. Now, that's great. We can see all of turtle diversity within this crown clade. But if we want to understand how these strange anatomical conditions came to be, we need to consult, well, we need to go deeper into turtle evolution onto a region of the tree that we call the stem lineage, which is a product of extinction. And what do you know? There are fossils down there. So for paleontologists, I'm largely a paleontologist. This is where a lot of our work happens. So you have extinct lineages of turtles branching off here that died off and that aren't part of the main extant radiation of turtles. Now, what's important to note that is often not very well communicated by science media is that these fossils never represent ancestors. We would be being scientifically dishonest if we were to say that they represented ancestors, because if we're making that claim, what we're saying is that we have overcome the preservation bias of geology and evolutionary time and found the exact population of these early turtles that essentially sat like right there. And of course, the rock record, the geological record, is highly biased. Not all of the rocks from every time period are preserved. And so to be safe, what we say is that these are close relatives, but actually represent their own lineages and are not direct ancestors. It's often not very well talked about in like popular science, things like that. So we can go over a little bit of how much has happened in turtle paleontology, because we found a lot recently, past 20 years. So the youngest, or the most crownward, the most closest to modern turtles that we know of, is this animal called Proganachelles, which is from the late Triassic of Germany. And as you can see, it pretty much looks like a modern turtle. It has a shell, it has a plastron, the body plan is largely in place. And we have multiple specimens of this animal, all pretty complete. So there's a lot of data we can gather from these animals. However, if we're trying to understand how we went from a generic reptile to this body plan, which is extremely strange and derived, this fossil actually doesn't help us very much. It already has everything that we expect to see in a living turtle. So how would it help us to understand turtle origins? Well, this is basically all we had to work with for 100 years, actually. It's a pretty old, this fossil was found for a long time, which is one of the reasons why it's been so difficult to reconstruct turtle phylogenies, because this was our earliest turtle and it already looked like a turtle. So it's not super useful for that. So what we have here is a fossil turtle, a fossil early turtle, called Odontocheles, which is from the late Triassic of China, published by a group of Chinese researchers. And we see kind of like a melange, kind of an accumulation of turtle-like characters, but not the complete body plan. So this is useful for us in understanding which parts of that turtle body plan came first. So we have widened ribs, but they don't fuse together like they do in turtles that we're used to, in living turtles. But the plastron itself, if you see on, so B up in the top right, that's the belly side up of the fossil. And the plastron is basically fully formed. So that's cool in and of itself, because it means we know that the plastron evolved before the dorsal carapace, which is kind of cool to know. This animal also had a lot of teeth. Of course, no living turtles have teeth. So this would be the closest animal to living turtles that retain teeth, which is kind of cool. We know that turtles evolved from animals that have teeth now. And the next one going down is Eorhynchocheles, again from China, 8 million years older. And what we have here is we have the neurals, which are basically the vertebrae. They form the very midpoint of the dorsal carapace in living turtles. They have flattened like they do in living turtles, so we have that stage preserved. And the ribs are doing the axial displacement, so they're staying in the axial line and not curving around into the midline like they do in us. And again, we have now another one we have is Proganachelles, and this was a really important find. This specimen is extremely complete, and it shows some really, really early turtle features. This is from Germany. And what we see here is that the gastralia, so those belly ribs I was talking about that go on to form the plastron, are there. There's no plastron yet, but they're widened. So it looks like they're in this state where they might go on to form the plastron. The ribs are short, like they are in turtles typically. They're not curving around, not as long as ours are. And it seems to be this is where probably the sternum was lost, which is a bone that almost all other amniotes possess. And then we come to Unitosaurus, which is the earliest early turtle that we know of. It's from the Permian of South Africa, almost 260 million years ago. And Unitosaurus is really important as the earliest turtle. It records the earliest transformations on the turtle lineage prior to the divergence of living turtles. So we have the fewer vertebrae. Seems like that was a very early transformation. Unitosaurus has it. It also has the broadened ribs, but they are still, it's kind of difficult to see, but in this fossil here, you can see they are curving round. They're not sticking out. So Unitosaurus had the curvature in the thorax. And really importantly, Unitosaurus has a preserved skull. And what we can see here is it is actually a diapsid. So for all those years where we couldn't find any kind of cranial evidence, which is morphological evidence that they diverged from within diapsid reptiles. I don't know if the mic still works. Okay. I can just take it off. Mic's off, I guess. Yeah. Because it preserves this diapsid condition, which is the first real morphological evidence that they diverged inside reptiles. All reptiles are diapsid. They have the two. So one of the principal questions that our lab is trying to get at, or one of the research areas that we're trying to get at, is to understand exactly how all of these shifts in anatomy occur over time in a biological way. I mean, the fossil record is limited in that, the fossil returns to rock. It's hard to conceptualize these animals as living, breathing organisms. So one of the ways at least we conceptualize the study of organisms in our lab is when we look at an organism, we call this the phenotype, aside from the genotype, which means the organism is the phenotype, and it's under the influence of two main creative forces. The first one being ontogeny, which is basically the individual lifespan of that animal all the way through embryological development to the turtle that we see walking around or in that box. And the other one is phylogeny. Basically, this is the influence of its evolutionary history. Animals are the product of their evolutionary history, and these two creative forces interact to form what we call the phenotype, which is what we study in turtles, the turtle phenotype. Now, in the past few years, something that's become quite obvious to us evolutionary biologists and has been pretty influential on the field is the idea that embryonic development itself evolves. What we usually think of evolution is that we have animals moving around an environment and they are selected for or against based on mutations and things like that. But what we've realized relatively recently is that the process of embryological development itself can evolve. And that actually it's through the evolution of embryological development that most of the diversity that we see amongst animals comes from. You change the way an animal develops, you change the way that it looks, you change the way that it operates in its ecosystem. And for people interested in evolutionary development in turtles, it's a good time to be working. One point being that we now have lots of people have been out and collected turtle eggs and characterized and described the way in which they develop from an embryo all the way through to a turtle. More and more turtles are having their genome sequenced, so you can start to get at genetic questions of how the turtle body plan came about. And also, and we can use those to plan experiments regarding genetic material of turtles, how the genes are affecting the way that turtles look. And one thing that turtle embryos have that no other amniote embryos have is this thing called a carapacial ridge, which is basically, you can imagine, you can't imagine being an embryo, but if you had a ridge, basically of skin that went all the way from your lower arm down to your leg, that's what we call the carapacial ridge. And it's been known about for a long time, but what it's actually doing in turtle development has not. Recent science, though, has characterized this region as a really important center for genetic activity. So there's a lot of jargon, you don't need to read all of the labels, but essentially this is a slice of a turtle, almost if you slice it through like this. And so this is the rib coming out of the vertebrae, and in turtles, they're axially arrested, so they stay up at the axial level and enter this carapacial ridge. And people have done genetic studies of that ridge, and it seems like it
might actually be the structure that is driving potentially a lot of these rib-related structural characteristics. And if we compare that to something like a chicken, if at the normal rib condition and the ribs start to develop out of the brain, the body will likely be enough. So this is a picture rich. Now, Paul, I'm going to hand over to Paul, who has been working with us for several years now. And Paul is really involved hands-on, doing a lot of these experiments and learning these techniques. So it's best that he speaks to you about.
I try to turn the mic on because people on Zoom are saying off. Cross your fingers.
It's working? No. Yeah. Let's try. Nothing. Nothing. It's coming over Zoom? All right. So we'll keep it on then. All right. So as Will alluded to, development is very important in making a turtle a turtle. And there's a lot of really unique developmental processes that happen that are unique to turtles. And now, what is driving that unique development are genes. So one of the big questions that we're after as a lab to try to answer, how does a turtle become a turtle, but also answer how other unique morphology, so other unique species come about, is linking genotype, which are, again, as Will talked about, the genes. So what are the genes that are expressed and when are they expressed? And how do those genes cause the physical traits we see? So how do these genes cause all the unique or the unique structures of life, and for us, the turtle shell? So specific to the turtle body plan, there's a couple areas of interest that we're really looking at as a lab. One of these is, as Will just talked about, the carapacial ridge. Another structure is the plastron. That is that ventral or the belly shell of a turtle. That's, again, very unique to turtles. And then the carapace. So that is dorsal shell or the back shell that's made up of the ribs and the vertebrae. So there's a couple techniques that we use to link these two and explain how do the genes and what are the genes, how do those cause the unique structures of turtles. And those are our genotype or our gene expression techniques, which are bulk transcriptomics, spatial transcriptomics, and then we have our visualization technique. So this is how do we see these structures physically, and this is histology and 3D modeling, and clarity staining and immunohistochemistry. And then as a lab, what we're really trying to do is bridge the genes and then these structures, how do they come together? And we're using integrative computational techniques to bridge these two. And I know a lot of these terms are pretty complex and you probably haven't heard them before. So we're going to take some time to dive deeper into what each of these techniques are and how we're using them as a lab. Now, before we get into these techniques, we have to acknowledge, what are the specimens we use? Where do we get them from? So we use reptiles as a lab. So we collect turtles, alligators, and chicken. And of course, we have to go get these embryos, so these eggs. And this makes for a really fun time as a lab because we all go down to Louisiana together and we go and pick up the eggs, drive them back as fast as we can to Baltimore, incubate them, and then extract the embryos that we need. So we can see Will's up here holding an alligator, and here are some eggs we have in an incubator, just some chick eggs, but we incubate all types of reptiles. And now this first visualization technique that we use, so again, how can we visualize these structures in development, is something called clarity and immunohistochemistry staining. And what this process is really doing is we take our embryo, so we use Strachemi scripta or the red ear slider. This is just a common pond turtle that's pretty prevalent across the east coast of the US. And what we do is we take this embryo and then submerge in a bunch of different chemicals that make it clear and transparent. And now with this clear or transparent embryo, what we can do is we can attach these protein markers called antibodies. So these antibodies will bind to a tissue of interest. And since our embryo is completely clear, we're able to visualize one single structure and one single tissue very clearly. And this is what this looks like. So this is a really cool image. This is something that we'll produce in our lab. And this is staining the muscles throughout turtle embryo. So there's 3 different turtles, and it's kind of hard to make out with this, so we'll throw on some eyeballs, so hopefully that'll help. You see the turtle that's in there, but this bright structure is the heart. You can kind of see the tail down here, and we're doing this technique, and what we're really showing is, imagine if you could only see the muscles in a developing turtle. So again, we talked how the muscles are present in the early turtle, but then they're lost over time. So right now we're able to visualize just the muscles. If we removed everything from the turtle but the muscles, how does that look as we go across developmental time? And this is what we're able to see. The next technique that we're using is just classical histology. We start with the same embryos that we'd start with clarity. So again, just our common pond turtle. It undergoes a couple of different chemical modifications, and we embed it into a wax blocks. And this is called paraffinization. And then we use a special tool called a microtome to make very, very thin sections, 3 to 7 microns, and that's smaller than a single cell. So less than a single cell thick are the slices that we make. So that what we're doing is we're taking our embryo, cutting it super, super thin so we can see the internal anatomy. And then it produces this thing that's a histology slide. If you remember way back to like biology in grade school, you might remember using these kind of slides to look at stuff. We stain it with different chemicals that brings out different structures. And using a microscope, we can now see the internal anatomy of a turtle. And I know there's a lot going on, so just a couple things to remember for the last little 10 minutes of this is if you see blue, blue means cartilage. This is our developing bones or developing skeleton. So that's pointing to the ribs here, the vertebrae back here, and the sides. And if you remember back to what Will was talking about, we start with ribs. Over time, those ribs widen out. But early on in our embryo, we still have ribs. So it's not yet a fully formed shell. We still have the rib condition as an embryo. If you see pink, think muscle and nerves. So you can see in between the ribs, there's a little bit of muscle, not as much as what there was earlier. And these muscles are breaking down throughout the embryo. And since we do this histology on the entire embryo, we create thousands of histology slides. So we do the same 3 micron thin section of an entire turtle, and we stack them just like layers of a cake. So we put each histology slide on top of each other, and then we can make this really cool video. So in these 2 videos, what we're doing is we're traveling through an embryo at a cellular level. So we can see the entire internal anatomy. Here in the turtle, we can see that's the spinal cord going through. On the left here, we see we have an alligator head. Remember, blue, think skeleton. Red, think muscle and nerves. And we can see that, you know, in this early turtle, there's still a lot of that pink, that red color. There's still a lot of muscles. It hasn't yet lost these muscles, which is really important in making the turtle shell. And then the next thing that we do is we're implementing AI and deep learning to help us with this goal as a lab. So if you remember, we went through this histology slide. We talked out blue means skeleton, pink means muscle. We're now using a computer to do that for us and doing it much better than what we can do as humans. So if we took that video of the alligator head, we stopped that alligator video at a certain point, just take a screenshot. This is what one of the hundreds of histology slides look like. Now we feed this into a computer, and the computer is going to tell us what every single different tissue is in this image. So we train it with a different modeling system, and it'll pick out what all the different tissues are, and this is what the computer will output, which is really cool. And now this is something that is very new to not just the field of developmental biology, but medicine. So this is the newest advances in medicine, and what they're doing in pathology is what they're doing is taking these histology slides and making 3D models of a tumor, of different organs. So this is published work from one of the collaborators that we work with to help us with our software. And this is a tumor, but we can see right now we have a 3D structure, and we can pull out every single different tissue in that 3D structure. So we can visualize now a turtle embryo in 3D at a cellular level, which is something that no one has been ever able to do for embryology, yet alone turtles, which is pretty incredible. And again, what does this mean we can do? That means we can pull different tissues out. If we want to look at just the skeleton that we say is really cool in turtles, we can tell the computer, show me the skeleton. If we want to look at just the muscles, we can say, show me the muscles. We can pull out these tissues in isolation and look at them at a 3D level, turn around, spin around, and see what they really look like. So those were our morphology techniques, our phenotype techniques. How do we see the structures? Now there's a bunch of techniques that we use to understand what are the genes driving that unique structure. And the first one is called bulk transcriptomics. So what we do is we'll take our turtle embryo, we'll remove an area of interest, and then we'll send it off to a core. This core undergoes a bunch of reactions, and they'll tell us all the genes that are expressed in that little tissue sample. Now, this is different from spatial transcriptomics, which is the next technique that we're also using in a lab. So while bulk transcriptomics tells us all the genes in that tissue, what spatial transcriptomics will do is it'll produce a histology slide just like the same way we do. Only instead of staining it, we send it to a core, and the core will tell us all the genes that are expressed on that slide. So what that lets us do is we can look at the structures here. We can say, what is that unique structure we see? And now we can link it to what are the genes that are expressed at that same location, which lets us know what are the genes driving this certain structure. And now what we're doing as a lab are integrating these methods. So we're taking the morphology and the phenotype characters, so that clarity, that histology staining, or incorporating that with this new generation of transcriptomics and gene expression techniques. So one of the things that we're working on is that we'll do histology slides of the entire embryo, like we talked about, but every couple slides we'll save it. So we'll pull that slide aside, and instead of staining it, we'll send it off to a core to do that spatial transcriptomics. And now what that lets us do is that lets us make a 3D model of our embryo, and we can see what are the unique changes in a turtle in 3D, pinpoint exactly where that's happening and when that's happening, and link that to the genes that are being expressed, because we have that slide. So we're able to overlay all the genes that are being expressed in a turtle embryo with what is the unique morphology. So what are the unique structures that are forming in that embryo, which is a really, really powerful tool and something that's like on the cutting edge of medicine, and nobody's been doing this for developmental biology, yet alone turtle research. So using these methods, what are some of the things we learned about turtles? Well, Will talked about the two main modifications to the turtle ribs. If you remember, instead of curving around, they go out flat in their back, and then they expand out to form the shell. Well, we knew those were happening for a long time, but we didn't know when they were happening or how they were happening. Using these techniques, we're able to visualize what exactly this looks like. What does it look like for the turtle shell to form during development? And then we can also figure out when this happens. What's the exact timing that these events are occurring, so then we can study it in even more detail. And since we make the whole 3D model, what we're able to do is we're able to see what's happening in the middle of the shell versus what's happening at the end of the edge of the shell. So that gives us a sense of what are these transitions looking like in space. So we're able to see how this turtle shell, how the turtle body plan forms not just over developmental time, but throughout space as well, which is really powerful. And we'll just focus in on two structures for the remainder of this talk, and those are the ribs and the muscles, both very important in the formation of the shell. And what we can see is that the ribs start out very densely nucleated. Lots of cells make up this rib. And then throughout time, as the embryo develops, we see more and more blue start to form in the staining. Now, this blue signifies that this cartilage is maturing. Blue means cartilage. That means this shell and these ribs are maturing throughout time. These white pockets are called lacunae, which means we've reached mature cartilage, so to say. So we're able to pinpoint when exactly this happens. And then what's really cool is at this stage, stage 21, so we're able to pinpoint this timing. What we see here is this smooth rib that we saw earlier is now bumpy. There's little spikes that are coming out of it. What this is that's that beginning of the rib expanding out wide to form the shell. So we're able to pinpoint at this exact time, this is when the ribs start to expand out to form the turtle shell. And this is something that we haven't been able to do before. We knew these processes existed, but we didn't know when exactly they occurred. Now, the next thing that we want to look at is the muscles. So we said that as these ribs expand out, the muscles disappear, which is one thing you can see. So we can now document how these muscles break down. Again, we knew they broke down. We didn't know how and when they did, and now we're able to visualize it. We see early on, we see lots and lots of muscles. So these dots are the ribs. In between the ribs, we have lots of pink. Pink means muscle, lots of muscle. Then we see how exactly these muscles break down, where they get less and less fibers, and eventually they get very, very small. And at the end, there's barely any left. There's just a couple remnants of junk, so to say, of muscles that are broken down that we can see in this pocket right here. So we can pinpoint the exact timing of when that occurs. And we can see that there's definitely a relationship between how these muscles break down and how the skeleton forms, which is something that people haven't really considered for turtles. So to summarize what we've learned in this talk, we went over with Will, what is a turtle? What are the unique structures that make a turtle, and what makes it the structures that we all love? We also described what the fossil record of turtles look like. We went through all those fossils, if you remember, how we got to our modern turtle. What do the old turtles look like? And they're pretty cool. We also talked about how this turtle shell and the unique turtle body plan comes about during development. We learned that development is the key. So whenever you think, how did the turtle become a turtle? Think it all happens in development. And then we briefly went over what are the new techniques. These aren't just cutting edge for developmental biology and turtle research, but this is cutting edge for all of science right now and for medicine. And we learned how can we apply these techniques to answer questions about turtles. Well, thank you for listening. That's all the time we have. And if you guys have any questions, feel free to ask.
Like, did it start, it was basically like, basically comparable to like the German and the fetus?
Yeah, so it's a structure that incorporates, yeah, you see it first in the feminist, yeah, it's an external structure.
And then usually like the last beyond.
Yeah, pretty much. So that structure defines the periphery of the shell. That's where the ribs embed and the plastron goes up to.
Even the adult turtle, that area is still considered the parafacial rib.
It's we probably call it a remnant. It's not really, it's not an embryonic structure anymore. It's now made of ossified skin, which is the surface of the shell, superficial surface of it. But yeah, it's there. It's the peripheral band.
I was gonna link it in here, but what lab are you guys involved with?
Yeah, so we're both at Johns Hopkins. We're both in the Center of Functional Anatomy and Evolution at the med school. I'm also part of the pathology department. So a lot of these techniques that I'm like, these are cutting edge medicine techniques. We do the same thing with some of the labs I'm a part of, the study cancer and how organs develop. So it's the same techniques, only we're applying it to evolution and the turtles, which is something that is doing, which is really cool.
Most people can't justify it. It's kind of expensive.
It looks expensive.
Yeah. Well, we're very lucky to have the funding.
Anything else?
Development wise, with most hard shell turtles versus like say soft shell or greater extreme the leatherbacks.
Yeah.
What would be the key difference?
Yeah, so really the key difference there is that in soft shell turtles, if you go to the first slide, so if you look at a soft shell turtle, most of the ribs still meet together, like they do here, but it doesn't stretch all the way to the end of the shell. It kind of terminates halfway, and then there's like this splint of bone that comes out, and so the tissue between them isn't totally ossified in soft shell turtles. And soft shell turtles, of course, don't really form bony scoots like adult hard shell turtles. You know, when we look down on a turtle, we are looking at basically ossified skin with the ribs underneath it. So it's a combination of both ossified skin and the endoskeleton, so the normal. But everything normal.
Yeah, so the ribs widen, but they don't like fuse together. But the hard shell, they're also missing extra bones.
Evolutionarily speaking, with that, especially the leatherback, that would technically earlier than most of the hard shells, or secondhand lost it after.
Yeah, that's what's interesting. So you have soft shell turtles, you go to the ground. So we talked about those two groups that define modern radiation turtles. You have soft shell turtles both in cryptodies and polaridies. So that condition evolved independently twice. And they're kind of nested within turtles that are hard shell. So it's something that evolved. It's brilliant. Probably has something to do with genes that control ossification. We don't know yet. There's so many questions.
So you mentioned, there was an odd anomaly, odd thing of the shoulder blade and the front leg within, underneath the ribs. Can you